The heart is a hollow muscular organ that is somewhat pyramid in shape and lies within the pericardium located in the anterior mediastinum. Through its pumping actions, it serves a vital role in providing the necessary oxygen for survival of the different organs and tissues of the body. The walls of the heart are composed of cardiac muscles known as the myocardium. The myocardium is covered externally by the serosal epicardium and internally by the endothelial endocardium. Muscle cells called cardiac myocytes are composed of actin and myosin filaments whose arrangement gives the characteristic striated appearance of the muscle. Two prominent organelles can be seen on the cardiac myocytes. These are the energy-producing mitochondria and the calcium-regulating sarcoplasmic reticulum. During the excitation-contraction coupling of cardiac myocytes, an electrical action potential generated from the heart's pacemaker opens the voltage-dependent L-type calcium channel called the dihydropyridine channel located on the T-tubules. This allows the movement of small amount of extracellular calcium ions into the cell. The calcium that enters the cell then triggers calcium release from the ryanodine receptor located on the surface of the sarcoplasmic reticulum. This phenomenon is more commonly known as the calcium-triggered calcium release. The calcium released from the sarcoplasmic reticulum now binds to the actin thin filaments and initiates contraction of the sarcomere. At the end of the muscle contraction, calcium comes out of the myofilament. Some of them are actively brought back to the sarcoplasmic reticulum through the calcium pump, while others are pumped into the extracellular space. We shall now take a closer look at the action of the sarcomere during excitation-contraction coupling. Cardiac contraction is brought about by the close interaction of the actin-thin filaments and the myosin cross bridges. In a relaxed state, the troponin and tropomyosin covers the active site of the actin filament. This prevents the myosin head from attaching itself into the active site. During contraction, however, calcium released from the sarcoplasmic reticulum binds to a troponin C of the thin filaments, which then shifts the troponin complex together with the tropomyosin into the active conformation. This would now enable the actin to interact with the myosin cross bridges. The myosin heads, which contains myosin ATPase, then hydrolyzes the ATP to provide the requisite energy for the myosin cross bridges to pull the actin filaments to the center of the sarcomere. This initiates contraction of the cardiac myocytes. Here are some basic differences between the skeletal and cardiac muscle. Unlike cardiac muscle, skeletal muscle initiates its excitation-contraction coupling via myoneural transmission. This will produce a conformational change in the dihydropyridine channels, stimulating calcium release from the sarcoplasmic reticulum. Cardiac muscles, on the other hand, initiates contraction via pacemaker potentials spreading through gap junctions. Because the sarcoplasmic reticulum of cardiac muscle is less extensive, the intracellular calcium supply for contraction is less. Cardiac muscle thus relies on extracellular calcium for contraction. Heart failure occurs when the heart's ability to pump blood has been weakened by disease. When the heart fails to pump efficiently, the flow slows down. This causes the blood returning to the heart through the veins to back up. Some of the fluid in the blood is forced out through the thin walls of small blood vessels into surrounding tissues. Here, the fluid piles up or congests. Patients with heart failure often present with ventricular hypertrophy. Digoxin, a form of digitalis, is usually given to strengthen the action of the heart muscles. Cardiac glycosides like digoxin inhibit sarcolemmal sodium-potassium ATPase pump, thereby increasing intracellular concentration of sodium. The elevated cytosolic sodium reverses the direction of the sodium-calcium exchanger and will secondarily cause intracellular calcium to rise. This is referred to as positive inotropic effect. The increase in calcium results to an increased force of cardiac contraction and is beneficial to heart failure patients.